Hi everyone, welcome to my channel. Today we are going to see about the basics of DNA. In last session we have covered that how DNA has been compactly folded and condensed in form a chromosome. In this video we will see about the what is DNA and what is the structure, what is this chemical compound. Okay, so what is DNA? DNA is nothing but the deoxyribonucleic acid. So let us see what are this chemical composition. As we see about the chemical composition, first chemical composition is DNA is made up of sugar molecules, phosphoric acid and nitrogen bases. As we see in sugar molecule is always a pentose sugar. In this video, as we covering the DNA and also we will cover RNA also. Okay. So sugar molecules are pentose sugar because in this compound sugar molecules contains 5 carbon compounds. Let us see. This is a sugar molecule compound in which 1, 2, 3, 4, 5 carbon compounds is present in the sugar molecule. Why I have drawn 2 sugar molecule means in one sugar molecule this is present in RNA and this sugar molecule is present in DNA. I will explain it because in second carbon position in RNA it's called as ribose sugar due to OH is present in second carbon compound. In DNA it is known as deoxyribose sugar in which the second carbon compound has H only, absence of O molecule. So that's why this is known as deoxyribose sugar. Oxy, D means elimination of oxygen, so ribose sugar. So these two compounds are pentose sugar due to 5 carbon. Pentam is 5, so 5 carbon compound contains this sugar. So next thing is phosphoric acid. DNA also contains phosphoric acid, one of its chemical composition. Its chemical structure is H3PO4. It makes the DNA acidic in nature and also makes this negatively charged molecule as we see in the last video. And finally we will come about the nitrogen base. Nitrogen is the most important thing which makes the structure of a DNA. Let us see. Nitrogen bases are of two types, that is purines and pyrimidines. What is purines and pyrimidines means? Purines is a two-ring compound structure and pyrimidine is a single ring compound structure. In pyrimidine, nitrogen base is divided into adenine and guanine, and in thymine nitrogen base is divided into, and in pyrimidine nitrogen base is divided into thymine, cytosine and uracil. But this uracil is not present in DNA, which is present in RNA. It replaces thymine in RNA. In RNA, thymine will not present, only uracil only present in place of thymine. Okay, and this is the composition of DNA, how it has been attached. Let us imagine this is a sugar molecule, pentose sugar, and this is a nitrogen bases in which adenine, guanine, thymine, cytosine and uracil, anything must be, and one phosphate molecule, phosphoric, that is phosphoric acid or phosphate. This pentose sugar may be ribose or deoxyribose. If it is ribose, it will be RNA. If it is deoxyribose, it will be DNA. Okay. Let's see about the detailed structure of nitrogen bases. As we told that nitrogen bases are divided into two categories, that is purines and pyrimidines. In purines, it is two ring structures, and pyrimidines, it is two
single ring compounds.
In purines, there is adenine and guanine, and in pyrimidines, it is divided into cytosine and thymine and uracil. In purine, this is the basic purine structure in which adenine, in this place, in sixth position of this compound, in place of H, NH2 is placed, and in guanine, in sixth position of compound, in place of H, double bond O is present and second carbon NH2 is present instead of H. Whereas in pyrimidines, in cytosine, this is the common pyrimidine structure. There are single ring structures. Whereas cytosine, in place of fourth carbon, NH2 is present. Whereas in thymidine, in place of fourth carbon, double bond O, and in place of fifth carbon, CH3 is present. Whereas in uracil, in place of fourth carbon, double bond O is present. So this uracil is present only in RNA, not in DNA. In place of thymine, uracil is present in RNA. As we seen the detailed structure of nitrogenous bases, so let us go by step how this DNA has been formed, how this DNA structure will be. So next step, the important rule is Chargaff rule. Erwin Chargaff told that nitrogen base adenine will always binds with thymine or uracil. In DNA, adenine will always bind with thymine, but instead of DNA, if RNA will be present, means adenine is always binds with uracil. Okay, and guanine will always bind with cytosine. This two lines are known as hydrogen bonds. Hydrogen bonds. Adenine binds with thymine with 2 hydrogen bonds, whereas guanine binds with cytosine with 3 hydrogen bonds. And one more thing is, the ratio of adenine plus guanine, cytosine plus thymine is always equal to 1. Adenine plus thymine, the ratio of adenine plus thymine and ratio of guanine plus cytosine is constant for a species. It may vary to species to species. For example, in prokaryotes, adenine plus thymine, this ratio will be less than 1, and this ratio in eukaryotes, this ratio will be greater than 1. In eukaryotes, there may be humans, mammals, etc. Prokaryotes, example, bacteria, like that. And then the important thing is the basic unit, that is the nucleotide. What is nucleotide means? That is a phosphate group plus pentose sugar, that may be ribose or deoxyribose, and nitrogen base. These are the nitrogen bases: adenine, thymine, guanine, cytosine, and uracil. This combines to form a structure or compound called as nucleotide. This is the structure. This is a very important structure which forms the DNA. This is the unit of a DNA or mRNA or RNA. And another important thing is nucleoside. Let us remove the phosphate group, then we call as nucleoside. Pentose sugar plus nitrogen base is called as nucleoside. Or let us say this nucleotide is also called as nucleoside plus phosphate. Okay, nucleoside is also called as nucleoside plus phosphate. This nucleotide is more important thing, which forms the repeating units, and combining of these units will form the DNA structure. Let us see the DNA structure. So as we see the structure of nucleotide, now let's see the detailed structure of DNA. How DNA has been formed, how DNA structure will be. So let us see. This is the DNA structure. DNA is an anti-parallel structure with a 2 strand. This is one strand, this is another strand. Okay. Let us see the detailed structure of a DNA. This is the detailed structure of DNA. As we see in the structure of nucleotide, one phosphate group and pentose sugar and one nitrogenous base. This pentose sugar, this is the DNA, therefore this pentose sugar is deoxyribose sugar, right? So as we see in Chargaff rule, adenine is always binds with thymine with 2 hydrogen bonds, and whereas guanine is always binds with 3 hydrogen bonds with cytosine. Like that way, this DNA is formed. This DNA is formed due to the connection between this phosphate group and the sugar linkage. This is known as sugar phosphate backbone. This backbone, this series of bonds are formed between sugar and phosphate groups are known as sugar phosphate backbone. Like that way, this another strand also will have sugar phosphate backbone. Without this, the DNA will be collapsed, and without this nitrogen bases, DNA will not be tightly joined with these two strands and form the two strands. Like that, DNA will have some names that is known as 3' end and 5' end, and 3' end and 5' end. As we told, the DNA is an anti-parallel in nature. Anti-parallel, DNA is anti-parallel. If this one strand will run in this direction and another strand will run in this direction, these 3' and 5' directions are denoted for the direction, or created for the direction. This 3' means this 5', this 3' means this 5'. Like that way, DNA will be formed like this. This 3' means this 5'. So this will be 3', another will be 5', right? And this is a nucleotide, and this 5' and this 3' will have, this 3' will have OH group, and this 5' will always have phosphate group. Like that way in that strand also. And this series of nucleotide, this nucleotide is a structural unit of the DNA, and this nucleotide will forms repeating units and form a polynucleotide chain. And this is one polynucleotide chain, and this is another polynucleotide chain. So DNA is also called as polynucleotide, right? And let us see this detailed structure, how this bond is formed, how this phosphodiester bond, and this bond is known as phosphodiester bond. The bond between sugar molecule and the phosphate group is known as phosphodiester bond. This is a very important bond which holds the structure of the DNA, which maintains the structure of a DNA. Let us see the detailed structure of phosphate bond, how this phosphate bond is connected with this compound. The formula is H3PO4, right? In this H3, 1, 2, 3 and PO4 molecules. And the sugar molecule, in base of the sugar molecules, it has in fifth carbon compound it will attach, and above the top sugar molecules, that carbon atom it will attach. So this forms the phosphodiester bond. This is an phosphodiester bond. Okay, this is the whole structure of the DNA. This DNA is always double stranded in nature. So uracil will present in, and one more thing is, where is, you may ask, where is the uracil? That is, uracil, I already told that uracil is present in mRNA or RNA only. Uracil is only the component which is present only in RNA. So uracil does not play a role in this compound, this structure. This base pairing, this is one base, this is another base. These two are known as base pair. So this base pairing is also known as, this A is always bind with T and G is always bind with C. This is known as complementary base pairing, also known as complementary base pairing. One correct thing will occur, that A is always binds with T and G is always binds with C with triple hydrogen bonds, and A is always binds with T thymine with double hydrogen bonds. This is a complementary base pair. So let us see this, what are the numericals in DNA and what are the distance and what are the lengths involved in DNA. Okay, so DNA is also known as double helix because due to this two strand structures, which is seems like a two snakes are combined together and spiraled in nature. Okay, let us see, the first one is diameter of the DNA. Diameter of the DNA is 20 angstrom or 2 nanometer in length. So diameter is 2 nanometer or 20 angstrom. This is the diameter of DNA. And what is the major and background is centrifugal, that is towards outwards the DNA is known as major groove. When the sugar phosphate backbone is towards the DNA, this is very close, or centripetal in nature is known as minor groove. This major groove and minor groove will combine to form as 34 angstrom in nature, that is 3.4 nanometer in a regular axis. When we draw an axis like this, from a center of a DNA is known as major axis, and this axis, when major groove and minor groove will form this 34 Armstrong or 3.4 nanometer. And this is a one turn. The DNA will turn, that is twisted in this form. That is one turn. We call this is one turn, and this is another turn, this is another turn, this is another turn. So one turn of DNA contains 10 base pairs. So this base pair means this is one base pair, this is another base pair, this is another base pair, this is another base pair. That is, if adenine is always binds with thymine, guanine is always binds with cytosine. Like this is one base pair, this is another base pair, like that. So distance between these two base pair, this two base pair, what is that? This two base pair is known as 3.4 Armstrong. This is 3.4 Armstrong or 0.4 nanometer in length. And DNA is always rotate in 360-degree direction. That is, this spiral rotates in 360-degree direction. Okay, this is the most important thing in this structure. So these are the more important things in the structure of the DNA while we studies. And this is the end of the video. Thanks for watching. So in next video we will discuss about then what are the types of DNA and what are the nucleotides are present, and what are the names used in nucleosides and nucleotides for various DNA compounds. In this video we covered the all basics requirement of DNA. In this video we covered all these basic requirements of DNA. Let us recap that session. First, see that we have seen that chemical composition of DNA, in which chemical composition of DNA is divided into sugar and phosphate and nitrogen bases. So this we combine to called as nucleotide, and this without phosphate group, the sugar plus nitrogen bases is called as nucleoside, right? Or also called as nucleotide plus nucleus. Nucleoside is also called as, so let us look at nucleotide is also equal to nucleoside plus phosphate. Okay, and we have seen that structure of nucleotide, this one phosphate group and the sugar compound and this nitrogen base and sugar. So these repeating units will form the DNA, and these repeating units will form the one polynucleotide. The two polynucleotide will form DNA. That is double helix structure. Double helix structure. And we have seen that how this phosphate bond is formed, how this phosphodiester bond is connected to this sugar molecules, and this is known as phosphodiester bond. And this linked with sugar is known as, also called as sugar phosphate bond. Okay, so thanks for watching like this. If you want to subscribe my channel, please subscribe to my channel. Thanks for watching.